Hi everyone, welcome to my channel. This will be my first journal club video of this channel, and the whole point of this is that is for me to go through a paper looking at the figures in detail and to enhance my understanding of immunology to Hep C, just immunology and biology in general. So I've come across this paper in Nature Immunology, and it's a very interesting paper. It's not the easiest paper to read. It took me quite a while to understand the figures in here, and that's because they use really fancy statistical analysis techniques. But I mean, that's the whole point of it, is for me to learn something. So this paper kind of describes how in chronic Hep C patients, even after the clearance of the virus, the memory-like CD8 T cells, so the key word here is memory-like, so they're not classical memory CD8 T cells. So these memory-like T cells, which give rise to the exhausted CD8 T cells, even after the clearance of the Hep C virus, they still retain the expression of the gene signatures associated with the CD8 T cell exhaustion. So yeah, it's a very interesting paper, and it also allows me to understand the heterogeneity of the exhausted T cells. So they're not just one distinct population. So they're made up of different populations. So they're still classified as exhausted T cells, but there are many different kind of subsets or just population in general. So hopefully you enjoy my video. This is my first video, so please don't be too harsh. But hopefully as I upload more videos, I will improve my video editing and just my presentation skills. So you have to keep in mind that this is not my work, so there'll be bits which I kind of stuck. But yeah, hopefully you enjoyed this video and learn something from it. And if you've read the paper, or just let me know, and see whether we are making the same interpretation of the data in here. Yeah. Hi, good day everyone. In this video I'm going to go through this interesting paper on immunity to hepatitis C virus. Specifically, this paper looks at the exhausted CD8 T cells that are produced during the chronic hepatitis C virus infection. I'll start off with the brief introduction of the paper. So exhausted CD8 T cells are not homogeneous and are made up of distinct subsets. So as with many immune responses, so cells within the infectious site should not be seen as a homogeneous population, but are made up of different populations. And relevant to this paper is what you call the TML cells, or the progenitor memory-like cells that give rise to the terminally exhausted CD8 T cells. And the aims of the study was to examine the transcriptional profiles, the trajectory and fate of these progenitor memory-like cells during and after cessation of chronic antigen stimulation. And the methods that they use, or mainly use in this study, were the flow cytometry and cell sorting for selecting the relevant cell populations, and with those cell populations they performed single cell RNA sequencing to look at the transcriptional profiles of those cells, and they did the bioinformatics analysis on those cells. Hi, for this first result section, what they did was they isolated the PBMCs from patients with chronic hepatitis C virus before they were given the drugs and after they were given the drugs, versus those who spontaneously cleared the virus, so those who didn't need the drugs, the lucky ones. So the PBMCs were given the different antigens, so the conserved epitopes, the variant epitopes, and the HCV specific T cells, specifically the exhausted T cells, were analyzed by flow cytometry using the two markers, so the PD1, the program death1, which is a marker for activation, but it's also a marker for T cell exhaustion, and the CD127, which is an interleukin 7 receptor. And with these two parameters, among others, they distinguish three different cell populations. So the classic memory cells, the TML cells, which is the progenitor exhausted CD8 T cells, and which give rise to the TTE, which is the terminally exhausted T cells. So they look at the percentage of HCV specific cells in colored dots, so red and blue, for example. And then they looked at the frequency of these cells, which can be plotted in these graphs here. And first they looked at the TML cells, and what they found was that the TML cells were found at the highest frequencies in after the drugs were given, as shown by the two blue bars here. You can also see them in the facts plot as well on the top right hand corner, and this frequency and this cell population, the TML cell population, was also found at a high frequency in also in the chronic individuals, but only in response to the variant epitope. And surprisingly, when they looked at the cell count of this population, of the TML population, there were no significant difference, which indicates that the difference that you see here is not due to the number of cells, but due to the percentage of cells. But when they looked at the TTE cells, which are the terminally exhausted CD8 T cells in the bottom right hand corner of the facts plots, so here, 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 and here, and what they found was that only that this population, the TTE cells, were only found in those who were suffering from chronic infection, and only in response to the conserved epitopes. And what they found here was that both the percentage and the cell number were significantly higher than other patients, which suggests that the more time the patients have had virus in their body, the more time the TML cells can convert. So the TML cells can convert into the TTE cells, hence the higher the frequencies of these cells. And they also looked at the memory, the classic memory cells, and they found that in this scenario, only the spontaneous clearers have the highest levels of the classic memory T cells, CD8 T cells, both in the percentage and the cell count. So it seems that in the chronic individuals, instead of T cells converting into memory CD8 T cells, the CD8 T cells were instead converted into the memory-like exhausted CD8 T cells. And lastly, for this figure, they did the correlation analysis, and what they found was that the frequency of the TML cells was associated with the production of interferon gamma and TNF alpha, as you could see here. So in this figure, what they did was they wanted to determine the transcriptional profiles of these two population cells, the progenitor cells and the terminally exhausted CD8 T cells. And so they performed the FACS sorting. So they select these populations using flow cytometry based on the two aforementioned parameters, so the PD1 and the CD127. So they sorted the cells and performed RNA sequencing, and then they performed the bioinformatics analysis, and in this case they used GSEA or gene set enrichment analysis. And in this study, the positive enrichment score means that the gene sets or the genes are enriched in the TE population, whereas the negative score, as you can see here, is below 0. So the negative score means that the genes are enriched in the memory-like, progenitor-like population. So what they found was that using GSEE, they found that the TE population is enriched in the exhaustion signature, whereas the memory signature and the memory-like signature are highly expressed by the TML population. And that's also the same with the CD8 tumor-infiltrated lymphocytes in human in hepatocarcinoma setting, as well as the B16 melanoma in mice. But when they looked at the cytotoxicity, what they found was that the TE are enriched in these genes. So clearly, I mean, it's reassuring that these two populations express different gene signatures. So next, they wanted to determine the expression at the protein level. So these are at the RNA level, the 2A and 2B, whereas the D, they wanted to look at the protein level, and so they did the TSNE analysis, TSNE analysis, and you could see the two different populations here. So the one on the left are the terminally exhausted CD8 T cells, and the one on the right are the terminally the memory-like cells on the right. And you see, so they looked at 6 different proteins: TCF1, TOX1, the EOMES, BCL2, IRF4, and TBET. And you can see the scale of expression here, which can be seen in here. And you could see that for a lot of these proteins, namely the TCF1, TOX1, and BCL2, they're clearly distinct expression levels, as you could see here in the bar graphs. So the TCF1 is mainly expressed by the TML, whereas the TOX1 is mainly expressed by the TE population, whereas the BCL2 is mainly expressed by the TML population. I couldn't see any different in the expression levels in the IF4, TBET, and EOMS between the two cell populations. So basically, what this figure is saying in a couple of sentences is that the two cell populations, the TML and TTE, express different gene signatures. Yep. So for this figure, what they wanted to find out was to look into the relationship between the two population of cells, so the TML cells and the TTE cells. So what they did was they performed single-cell RNA sequencing on the HCV-specific CD8 T cells from chronic HCV patients, and single-cell RNA sequencing revealed that there were 3 different clusters. So there were, cluster 1, cluster 2, and cluster 3, and you could see that based on single-cell RNA sequencing, there were 3 different expression profiles. So you could see clearly that in cluster 1 you see a blue block here, and the orange red block here, and vice versa. In cluster 3 you see there's a red block here, which represents the different levels of expression. So some of the well-known genes associated with the function of cells were found here as well. So CCL4 and CCL5. So these are the chemokines. So you could see that from cluster 1, there's a low level of these chemokines, whereas as cells kind of differentiate into cluster 3, they upregulate the expression of these genes. And you could also see the granzyme A and granzyme K as well, and vice versa. So for cluster 1, it has a high level of interleukin 7 receptor, whereas cluster 3 express a low interleukin 7 receptor. So you could see them here, and the Pearson correlation also showed that the cluster 1 and cluster 2 are very similar. So as you could see from this figure here, is that the cluster 2 lies somewhere between cluster 1 and cluster 3 based on the expression levels. And so cluster 1 and cluster 2 are very similar, and cluster 1 and 3 are not very similar when you take into account the cluster 2. And there's this analysis technique that I've never heard of, and is something that I'm not familiar with. I'm just going to go with what the paper said. They use this analysis technique called the diffusion pseudotime analysis, and what they said was so there's a continuous dynamic from cluster 1 into cluster 3. So basically there's like a continuous differentiation of cluster 1 into cluster 3. That's like my take from the figure anyway. And it's the same with the expression of the genes as well. I think I've shown you this already. So I mean, like this figure here and this figure here are very similar, but this plot or figure looked at the expression levels over time. So you could see that over time the interleukin 7 receptor and BCL2 kind of decrease in the expression levels as the cells move from cluster 1 into cluster 3, and with coenzyme K and HLA-DRA, the opposite is true. So this is reassuring that the three clusters express different levels of these genes, and I think what this plot is showing is also the same. So some of the genes were downregulated as cells move from cluster 1 into cluster 3, but some genes were upregulated. Yeah, and as with figure 2 that I showed you previously, so cluster 3, which kind of represents the TTE population, which is the terminally exhausted CD8 T cells, they express quite high levels of the genes associated with T cell exhaustion compared to cluster 1, which represents the TML, the memory-like progenitor cells. And whereas with the memory, the opposite is true. So cluster 1 tend to express the genes associated with the memory cells, and cluster 2 tend to be somewhere in between. And interleukin 7 receptor, I think I've mentioned this quite a lot of times already. So cluster 1 express more interleukin 7 receptor than the cluster 3. So basically what the authors were trying to say in this figure was that the cluster 1, which represents the TML cells, gives rise to the cluster 3, which represents the TTE cells with an intermediate population of cells. All right, so for this figure, what the authors wanted to find out was whether cells from patients who were given the drugs, whether their exhausted phenotypes can be reverted back to the classical memory CD8 T cells. So what they did was they performed flow cytometry. So they took the cells from patients before they were given the drugs and after they were given the drugs in blue, and from patients who didn't need the drugs in green. So if you just look at the areas here, so there are 3 different clusters, right? And so they looked at 5 different markers. So the TCF1, BCL2, TOX1, EOMS, and T-BET. And so these are gated on just CD8 T cells. So as you can see, TCF1 and BCL2, there's a degree of overlap, right, in the expression of these 2 transcription factors. It is expressed at a high level by the resolvers, so those who clear the virus without any drugs, but it is also expressed to a certain degree by patients after they were given the drugs, but less by the patients before they were given the drugs. And that's the same with BCL2 as well. But in the case of TOX1 and EOMS as well, you could see there's a clear clustering going on. So TOX1 and EOMS are not expressed by, well, to a lesser degree anyway, expressed at a very low level, very low levels by the spontaneous clearers, as you can see here, but it is highly expressed by the cured HCV, and even before they were given the drugs. And TBET, it is expressed almost exclusively by the chronic patients before they were given the drugs, as you could see here. So next they get it on the PD1 and CD127. So now they get it on the TML cells now, and you could see here in green there's like a north-south divide. So the resolvers, so the spontaneous clearers at the top, and the before and after the treatment at the bottom. So, and it seems like the cells of the before and after the treatment, they kind of intermingle, so there's not much difference in terms of the expression of these phenotypes. And so it seems like even after you clear the virus, the memory cells in chronic Hep C patients still retain what the authors called a molecular scar. So they couldn't shake some of these markers off even after clearing the virus. So, for example, you see, TOX1, they couldn't shake off the TOX1 expression, same as EOMS. There are some attempts to convert back into a classical memory cells, as you could see here, BCL2, so there's some expression, but it's not high. But I mean, the main message from this figure and from this paper as well is that, even after clearance, these T cells retain the molecular scar. And if you were to compare, using the gene set enrichment analysis, if you were to compare the resolved HCV memory cells versus the cured HCV, so patients who were given the drugs, the cells from those who were given the drugs, so the exhausted T cells, still retain the exhaustion markers even after the cure. And looking at the different in the gene expressions, so there were 1,419 differentially expressed genes between the two types of patients. So the genes on the left hand side of figure 4D, they were upregulated in the patients who were given the drugs in the CD8 T cells that were given the drugs. And on this side, so on the right hand side, these are the genes that were upregulated in the spontaneous clearers, the lucky ones. And these are some of the biological processes that were differentially expressed between the two, and the two patients. So you have the different Go terms in blue, different KEGG pathways, and different the biological process. And in E as well, so there are different expression in the Go terms, and I think some of them are the leukocyte differentiation and the regulation of T cell activation and response to the virus and the apoptosis. And you could see from this heat map here, you could see there's like a distinct block of genes that were differently expressed between different cell populations in different patients. So here, so this area here, so in green represent the cells from the spontaneous clearers, and in blue, which is here, sorry, the mouse is not very good. So here and here are from those who were, who were given the given the drugs, and this block here is the one before they were given the drug. So there's some, similarities between, cells before and after giving the drugs, but they are very different in terms of it's like heaven and hell here. So yeah, and there's not much to say. I mean, the figure speaks for itself. So you see a block of gene here and here. All right, thank you. All right, so in this figure, what they wanted to find out was they wanted to look deeper into the fate of the TML cells. So what they did was they took the cells from patients with chronic Hep C infection, and they took the cells before and after the patients were given the drugs. And the single-cell RNA sequencing found that there were 3 different clusters of cells. So there were cluster 1, which is shown in pink or light red, and the cluster 2 in the dark red, and cluster 3 in the crimson. And hopefully we can come to the same conclusion that using this bar graph here is that after you treat the patients with the drugs, you lose this population here. So you lose the cluster 3, which author stated that is the exhausted T cells, because they express the exhaustion signature. So you treat the patients, you lose this population, but you still maintain a little bit of the cluster 1 and cluster 2 here. And these cells can be classified into according to their interleukin 7 receptor gene expression, both at the mRNA level and the protein level here. So you could see the high expression here, I would say, using this scale. So you see a high expression here, you see intermediate expression, and then you see low expression of interleukin 7 receptor, and the FACS plot also show that as well. So you see high, intermediate, and low. And what they also showed was that you lose so when you treat the patients with the drugs, you kind of lose the CD127 low population, which I believe is which belongs to the cluster 3. So you lose pretty much almost completely. Then you have more of the CD127 intermediate population. There was no, so there was no increase in the CD127 high population. So basically, what figure 5A and C showed was that only the cluster 1 and cluster 2 were maintained over the course of the treatment. So they performed this differential expression analysis, and what they found was that the cells from cluster 1, before and after the drugs, there weren't that much different. So the cells are pretty much the same in terms of their expression profiles. There's only like 18 differentially expressed genes, and similarly cells
that cells that were classed as cluster 2, there weren't no, that there weren't much difference in terms of their expression profiles before and after the treatment. And they also use this like really fancy algorithm that I don't really know much about, but I mean, I hope I am right. So the more red it is, using this scale here, so the more red it is, the more similar the cells are before and after the treatment. So you see that in this area here, cells were similar. There were some cells that were, unique to patients before and after the treatment, but I think all in all, I think a lot of them are pretty much the same. They looked at the expression of TCF1, transcription factor, and the TOX1. So they looked at the different CD127 populations. So these are CD127 high. So these are the CD127 high population. As you could see, using the bar chart here as well and the facts plot, look at the mean fluorescence intensity. There was no difference at all. No difference before and after in the expression of TCF1 before and after the treatment. And similarly, the TOX1, there's no difference as well. And they also looked at the T cell receptor clonotypes using T cell receptor sequencing, and what they found was that before and after the patients were given the drugs, the number of pretty much the clones of the T cells. So the number of clones were maintained throughout the course of the of the treatment, and there were some T cell clones that were shared between the patients who cleared to. So there were some clones that were shared both before and after the patients were given the drugs. So what this figure is saying is that even after you treat the patients, you lose this population of cells, the C3, the cluster 3 population, which represents the exhausted CD8 T cells. You lose that population, but you still maintain the cluster 1 and cluster 2 population. And the cluster 1 and cluster 2, in terms of that gene expression profiles, are not different before and after the treatment at all, which indicates that even after you clear the virus, with the drugs, the cells still maintain the some exhaustion markers, and they just can't shake off these expression profiles, even when the antigens are cleared from the system. Yep, so in this last result section, the figure 6, what they wanted to find out was how the recognition of antigens influence the expression of these so-called molecular scars. So what they did was they selected the CD8 T cells that were specific to the conserved epitope, and the CD8 T cells were specific to the escaped epitopes, and cells from the spontaneous clearers. And you can see, three different clusters here, 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 and here. And hopefully what you can see here is that the this area here, which is occupied by the cells that were targeting the escaped variant, and they kind of overlap with both the populations of cells. So this population of cells have a similar expression pattern to both the cells of the spontaneous clearers and cells that targeted the conserved epitope. For TOX1, let's just come back to BCL2. BCL2 tend to be highly expressed by the spontaneous clearers, and cells from chronic Hep C patients that targeted the escape variant also express a relatively high level of BCL2, but at the same time, there are some cells that express relatively low levels of BCL2. So it's quite a heterogeneous population. TOX1, you could see clearly that the cells of the spontaneous clearers don't really express the TOX1 transcription factor, and TOX1 is highly expressed by these CD8 T cells from chronic patients, and same with EOMIS, and it seems like Tibet is expressed almost exclusively by the CD8 T cells that targeted the conserved antigen. So what they wanted to find out was how the TML cells also cluster. So the top in figure 6A was just CD8 T cells. This one they want to dive deeper into these progenitor memory-like cells, and again they performed a flow cytometry and T-cell plot and looked at some of these markers, among other markers. So this area again, so in green, is mainly occupied by the cell of the spontaneous clearers. The crimson is cells that targeted the conserved epitope, and the bright red is the one that are occupied by cells that targeted the escape variant. And hopefully what you could see here, there's again there's some clustering. It's very clear with BCL2. So BCL2 again tends to be expressed highly by the cells from the spontaneous clearers, as shown in this area here. TOX1 again, there's like a north-south divide. So cells from the chronic patients tend to express TOX1, whereas the resolvers don't really express TOX1, or at a very low levels anyway. Maybe it's probably not correct to just draw a line here, because as you could see let me just change it to the laser. As you could see here, you could see so you could see some red dots here, which represent the cells from the chronic patients. So what you see here in red, these are the cells from the chronic patients. So please bear that in mind. But EOMES is mainly again, like EOMES is mainly expressed by the cells of the chronic Hep C patients, and a Tibet as well. So next, I want to look at the expression patterns of these cells. So in green, again, these are the resolvers, whereas in blue and so in blue and red are the cells that target the escape variants, whereas in the dark blue and crimson are the one that target the conserved antigen. And you could see, so if you just like compare, you know, the cells that targeted the conserved epitope versus cells from spontaneous clearers, you could see there's a night and day here. So there's like a low expression of these genes by cells of the spontaneous clearers, but for the chronic patients is the opposite. So the chronic patients express a lot of these genes, and vice versa. So these genes are highly expressed by the spontaneous clearers, but they are lowly expressed by the chronic patients. But the cells from the chronic patients that targeted the escape epitope, they tend to lie somewhere in between. Let me just change it to the laser. Yeah, so they tend to lie somewhere in between compared to these two cell populations here. So next they looked at the different in the expression levels of some genes, and what they found was that there were so they found 352 differently expressed genes, and some of the genes that are regulated in cells that targeted the conserved epitope were TOX1, CD38, and TNRF, SF9, and KLRG1. And they also found that in cells that targeted the conserved epitope, they tend to express the exhaustion signature compared to cells that targeted the escaped variants. So all in all, what this figure is saying is that yes, the the recognition of antigens does have an effect on the polarization and the imprinting of these so-called molecular scars. Um, but my take on this is that the cells that targeted the variant epitopes, because the patients were given the drugs, so they cleared the virus, maybe it could just be that the CD8 T cells didn't really have the time. So you see here, so the CD8 T cells didn't really have the time to fully express the molecular scars at a high level compared to cells that targeted the conserved epitopes. So here's a summary, which I've drawn a figure which depicts my understanding of the paper, of the study. So here we have a patient with hepatitis infected with the HCV. That person can be, lucky. Depends on the, on their immune system. That person could clear the virus, and the
effector CD8 T cells can become memory CD8 T cells, but in the case of chronic patients who couldn't clear the virus, so there's persistent antigenic stimulation, their CD8 T cells become what you would call the progenitor memory-like CD8 T cells, which can give rise to the terminally exhausted CD8 T cells. However, if this patient was to be given the treatment, so the DAA or the direct action antiviral therapy, the progenitor CD8 T cells in green here kind of like partially convert into the memory CD8 T cells, but at the same time they retain quite a lot of markers associated with the T cell exhaustion. And if there's a figure from the paper which kind of summarized the whole study, personally I would say is figure 4. So if you look at the CD8 T cells, so the TML cells, you would see that these cells, they even though the patients have cleared the virus, they still express markers associated with the T cell exhaustion, so like TOPS1 and EOMS, but at the same time they also express markers associated with the memory. So basically, you can't get rid of your scar